Okay, so welcome to this biology webinar session. We're going to be talking about all the sections in Chapter 20 in the biology textbook. And you're going to see some questions on the screen for this webinar, and they're just to follow along with. There's no way for you to submit your answers. I'll just give you time to think about them and then show you the correct answer. So if you are here live, you can ask questions. If you're on a computer, you click on the little speech bubble, and that'll bring up a place to type questions. If you're on a phone or a tablet, you click on the question mark icon, and that will give you a place to ask questions. So don't hesitate to ask questions. You're not going to interrupt at all. You can ask me to go back and repeat anything or go over anything again, and I definitely will. You can ask other questions about things related to this lesson. That's fine. Anything that you want to ask about, as long as it's relevant. So this chapter is about the germs, viruses, and bacteria and archaea. And we start with viruses. So we know that viruses make us sick, and so we kill a lot about killing germs. So many people assume that viruses are alive. But actually, a virus is not a living thing. It is made of proteins and nucleic acids. Remember that nucleic acids means either DNA or RNA. And those are important to living things, but the virus is not a complete cell. Those are the only parts that it has. And it can evolve over time, but the individual viruses don't grow and develop like living things do. And then here are two other reasons that viruses are not living things that your book discusses. Viruses can't reproduce by themselves. They need to use a living cell to do that. And some viruses can be crystallized, which is something that living things can't do. That's how they kind of first discovered that viruses were something different. So a virus generally has DNA or RNA surrounded by a protein coat called a capsid. And they may have a membrane around that, similar to a real cell. But on the surface of the virus are going to be proteins that can bind onto the surface of a cell and trick that cell into accepting the virus. Because of this, a virus can only infect a cell with specific proteins. So each virus can only infect a certain type of cell from a certain type of plant or animal or bacteria. Okay, so you can't usually have viruses that affect a wide variety of species because they're looking for specific proteins. Now, there are two possible types of viral infection, a lytic infection and a lysogenic infection. In a lytic infection, the virus injects its DNA into the cell, and it kind of just floats around in the cell, but forces the cell to make copies of the viral DNA and proteins. So the cell is manufacturing viruses. The cell makes new viruses until it fills up with viruses and bursts, releasing them to infect other cells. In a lysogenic infection, the virus injects its DNA directly into the host's DNA. It becomes part of the DNA of that host cell. And then the cell kind of just continues living and reproducing normally, and all of its offspring that it produces are going to have the virus DNA in them. And then eventually something triggers that virus, and it can then burst a cell and start infecting even more cells. It kind of moves into a lytic infection. We're going to look at the same thing here in a diagram. All right, so we're going to start here. So this is a lytic infection. The red DNA is the virus DNA. So it goes into the cell, and then the cell starts creating the pieces of the virus and then starts assembling them. And then at the end, we've got all these complete viruses made. And they pop out of the cell, and the cell is destroyed. And then the virus leaves the cell and finds a new cell to infect, injects its DNA in there, and it does that again. So the lytic cycle is destroying the cell because the cell is just being used to manufacture viruses. A lysogenic cycle is a little bit different. In the lysogenic cycle, the DNA of the virus just becomes part of the DNA of the host, and then the host just keeps reproducing and making new host cells that have the virus in them. Okay, so the cell split apart into two cells, and each cell has this chunk of viral DNA in it. So the lysogenic cycle isn't really destructive. Um, it lets the cell keep living, keep doing its thing, but now it's just also creating more viruses. And then eventually, it kind of triggers it to move into the lysogenic or the lytic cycle. So it goes from this stage into, okay, now all these cells that have viral DNA are going to start making viruses really quickly. Now, I was saying DNA a lot on the last slide, but actually most viruses use RNA instead of DNA as their genetic material. The common cold is a virus that has RNA, and the host cell makes RNA for it. And HIV is an example of a type of RNA virus called a retrovirus, where the virus has RNA, gives it to the host cell, and the host cell uses it to make DNA. All right, so before we move on to the next section, we're going to review a little bit about viruses. So again, there's nothing to click on. There's no way to submit this. Just think about your answers for a few seconds, then we'll go over which is right. So which of the following are true about viruses? And I'll tell you now that there's more than one right answer. So which ones are true and which ones are false? So viruses, do they have DNA or RNA? They are alive. They can evolve over time. Individual viruses are born, grow, and develop, or and they obtain and use their own energy. So which ones are true? Give you about 10 seconds to think about it. All right, so which is true? They have DNA or RNA. That is true, but they are not alive. So they have a couple of parts that real living cells have. They have nucleic acids, the DNA. They have proteins, but they're not an entire cell. So they have DNA, but they're not alive. The next one, do they evolve over time? Yes, we're going to talk about that, how they can change a little bit. An individual virus is born and grows up and develops like a living thing? Nope. Oh, those got backwards. No, they're assembled inside the host and that's it. They don't really change anymore. And they obtain and use their own energy. That's also not true. They require a host to do anything. And one more review. Are these statements true for lytic infections, lysogenic infections, or both? So here's our statements. The virus can use DNA or RNA. The host cell makes and assembles new viruses. The virus DNA becomes part of the host DNA. The host cell fills with viruses and bursts, and the host cell keeps living and reproducing normally. So each one of those is either lytic, lysogenic, or both. Again, I'll give you about 10 seconds to think about your answers. Okay, and we'll go through them. And if you didn't know these right off, that's okay. I mean, first of all, it's kind of a long question to be thinking about in such a short amount of time. And also, we just reviewed it pretty quickly. So just go back in your book and study the difference between the two of them. So here's our things. The virus can use DNA or RNA. That's both. Any lytic or lysogenic infection, it doesn't matter whether they're using DNA or RNA. The next one, the host cell makes and assembles new viruses. That's a lytic infection. The next one, the virus DNA becomes part of the host's DNA. That's lysogenic. The next one, the host fills with viruses and bursts. That's lytic. And the last one, the host cell keeps living and reproducing normally with the virus inside of it. That is lysogenic. Okay, so you could always pause if you need to write any of this down. You could review, you can come back to it later. But definitely study the difference between lytic and lysogenic infections. All right, chapter 20, section 2. There are two types of cells. There's eukaryotes and prokaryotes. We learned this earlier. Um, okay, sorry. So eukaryotes are cells that have a nucleus. So that's these over here. They have a nucleus where their DNA is stored. And prokaryotes are cells that do not. So their DNA is just kind of floating out in the middle there. And we're talking a lot about prokaryotes in this chapter. Prokaryotes are the ones that don't have a nucleus, and there's only two types of prokaryotes. There's bacteria and archaea. So I say only two, but that's there's two domains or two kingdoms. I'm sorry, two kingdoms that have prokaryote cells, which is bacteria and archaea. But there's lots and lots and lots of different kinds of both of those things. Eukaryotic cells, the ones that have a nucleus, that would be plant cells, animal cells, protists, and fungi. Those are all eukaryotes. And some other characteristics of prokaryotes, again, they don't have a nucleus. Their DNA is just hanging out in the cytoplasm here. They are smaller than eukaryotes, and many of them have a cell wall around their outside. So we're kind of familiar with bacteria. They're prokaryotes that live all around us. Some are harmful, some are beneficial. But what about this other word, archaea? What are those? Archaea are similar to bacteria, but their cell walls and cell membranes are made with different molecules. And that's such an important difference that they put them in a different kingdom. The other big difference is their DNA. The DNA of archaea is different than the DNA of bacteria. In some ways, it's a lot like eukaryote bacteria or DNA, but they don't have a nucleus. And the third thing is that archaea tend to live in harsh environments. Okay, so like hot springs and vents on the ocean floor, places that most other things don't want to live. Okay, in the structure and function section, we learned that there's a big variety in prokaryotes. They could be rod-shaped, sphere-shaped, or corkscrew-shaped. They could be stationary or move in a couple of different ways. And they have different ways to get energy, which we're going to talk about on the next slide. So on page 486, there's a chart showing ways in which prokaryotes can capture and use energy. We learned these terms back in chapter 3 and again in chapter 8. A heterotroph eats other things, and an autotroph makes its own food. Some prokaryotes are heterotrophs. They have to take in food from their environment. Some are photoheterotrophs, meaning they do a little of both. They use light to make some of their own energy through photosynthesis, but then they also feed like regular heterotrophs. Photoautotrophs just use light to make their own food. And then we have chemoautotrophs, which use energy released by chemical reactions to make their own food. So these are just basically four different ways that different prokaryotes can use to get food molecules inside their body to provide themselves with energy at some point. And by body, I mean one single cell. All prokaryotes are just one cell. But they could either make it themselves or get it from their environment, and depending on how they do it, we classify them one of these four ways. Then on page 487, There's another chart about energy released by prokaryotes, and this is how they actually get the energy out of their food. So the previous chart is about how they get the food molecules inside the cell, and this is about once it's there, how are they actually releasing that energy. So this is concepts that we discussed in chapter 8, talking about cellular respiration and fermentation. Cellular respiration uses oxygen. And some prokaryotes can only get their energy this way, through cellular respiration. So they require oxygen. And because they require oxygen, they are called obligate aerobes. Okay, so obligate means they need to, like think of the word obligated, something you have to do. And aero is air or oxygen. Okay, so these are obligate aerobes. They need oxygen to live. Some use fermentation to release the energy in their food, and they actually die if there's oxygen around. So those prokaryotes are called obligate anaerobes. Okay, they have to be somewhere where there is no oxygen. And then there's some prokaryotes that can actually switch between fermentation and cellular respiration, so they do fine whether there is oxygen or not. And these are called facultative anaerobes. Okay, now binary fission is the way that prokaryotes reproduce. It is really similar to other forms of cell division. The cell gets larger, copies its DNA, and divides in half, forming two identical cells with identical copies of DNA. Now, a problem with this is that there's no genetic diversity. So many prokaryotes have to figure out a way to make that happen. So they use what's called conjugation. Conjugation is just a hollow tube that forms between two of the cells, and they exchange genetic material between them. This allows the bacteria to share genetic information with other bacteria of the same species nearby. And we tend to think of bacteria as bad things. We call them germs. We say they need to be killed. And some are, but there are also many very helpful bacteria. Inside of your body are bacteria that help you break down food and make certain vitamins and do a lot involved with digestion. So you need that bacteria inside of you to live and be healthy. Photoautotrophs, remember they're bacteria or archaea that make their own food from sunlight. And that makes them the base of many food chains. They're where the food starts. So they provide the energy for many organisms to live. They also produce oxygen while they're at it. Bacteria compose dead things, which is really important. It gets rid of carcasses and wastes and returns the nutrients back to the soil so that other organisms can use it. If we didn't have bacteria doing that, we'd quickly run out of nutrients for living things and get a big pile up of dead things. So it's really helpful to have the bacteria around to do that. Another important thing they do is make nitrogen useful. So there's bacteria called nitrogen fixers that take nitrogen from the air that no living things can use except these bacteria, and they turn it into compounds that plants can use. So without them, the plants would have no nitrogen and couldn't live. So these nitrogen fixers are really important to make nitrogen into a form that plants can use to live, and then we can eat the plants and keep on living. And we also put bacteria to work for us in a lot of industry things. We use bacteria to make yogurt and cheese and other foods, to process foods like cocoa and coffee, cure meats, make some food additives, cleaning wastes out of water, and we use bacteria as factories to produce a lot of drugs, including insulin for diabetics. They're made by bacteria. So that's the end of section 2, so we have a bit of a review. So again, just kind of think about your answers, and then we'll go over what's correct. So which of these options are prokaryotes? There may be more than one answer. So prokaryotes include bacteria cells and archaea cells. Yeast are not prokaryotes. They're a fungus, so they are eukaryotes, so that's not right. And viruses are not alive. They are not cells, so they're not prokaryotes or eukaryotes. They are just things that are not living. And next question, which of these are not uses of bacteria? So three of these things are helpful things that bacteria do for us, and then there's one that is not. So which is not? So our options are decomposing waste. That is something that bacteria does. Digestive help. Yes, you have bacteria living in your guts that do a lot of digesting for you. Taking useless nitrogen from the air and converting it into compounds used by plants. Yes, we need bacteria for that. And the last one is the one that's not true. They do not help viruses cause lysogenic infections. Okay, bacteria are sometimes what's being infected by a virus. And section 3 is about diseases caused by bacteria and viruses. So we just talked about how some bacteria are helpful, but of course others cause disease. A pathogen is anything that causes a disease. So we're talking about bacteria and virus. Fungus can be a pathogen too. A host is the organism that's infected by the pathogen. And when we're talking about bacteria, bacteria can either destroy living cells or release chemicals that interfere with the activities of the host body. And virus can destroy the host cells, and it can upset homeostasis and also interfere with the activity of the cells in the host body. And remember, a virus usually attacks a specific type of cell in the body. On page 489 and 490 in your book, there's two charts about specific bacterial and viral diseases. That gives you a lot of information about each disease. So be sure to check those out and use those on your exam. Now there's a section about fighting the diseases and preventing them. So how to combat bacterial pathogens. The most important things are stay away from anybody that you know is infected. Try not to spread it yourself. Cover your mouth if you sneeze and things like that. And then to keep from spreading the germs to yourself and others, you want to make sure you're always washing your hands, especially before eating, before touching your face. You need to wash your hands with soap for 20 to 30 seconds, and that's enough to remove the bacteria from them. Now, it doesn't kill them, but it removes the bacteria from your hands. Same thing with washing surfaces, so countertops and tables and objects. Washing them with soap will remove the bacteria. It won't actually kill them, but it'll remove it. To actually kill the bacteria, you need a disinfectant. And note that many of them require the bacteria to be wet with the disinfectant for 30 seconds. So keep that in mind when you're using things like disinfectant wipes, like Clorox wipes or Lysol wipes. A quick swipe of that disinfectant wipe may not be enough to kill the bacteria because the surface needs to stay wet with it for 30 seconds. So you might need to keep using fresh wipes so that it's really wet so that the surface stays wet for that long. Okay, temperature is something that'll kill them. So boiling water is hot enough to kill bacteria. If you have objects or surfaces that can be put into boiling water for 30 seconds, that ought to kill them. When you're cooking food, you want to follow the guidelines for food to be sure that food is cooked enough to destroy bacteria. Meat and eggs always have temperature guidelines, how hot it should be inside of it to make sure it's cooked all the way to kill that bacteria. And cold temperatures stop or slow the growth of bacteria. So refrigerators are kept cold enough to stop bacteria from growing. It doesn't kill them, but it prevents them from spreading. Bacterial diseases are treated with antibiotics. These are drugs that prevent the bacteria from reproducing without harming the host's body as well. I want you to note that these only work for bacterial infections, not viral infections. So if you go to a doctor for an illness, pay attention to what it is and whether it's an illness caused by a virus or bacteria. A viral infection, such as the flu, common cold, or chickenpox, cannot be treated by antibiotics, but a bacterial infection, such as strep throat, that can. And then some viral diseases can be treated with antiviral drugs, which attack proteins that are on the virus cells but not the host cells. But if your doctor tells you you have a viral disease and there's no way to treat it, and unfortunately you just have to go home and get over it, that's really unpleasant news. Nobody wants to hear that, but sometimes that's the case. So don't try to demand antibiotics because if it's a viral disease, that's not going to help. It's just going to make things worse. So when we're talking about diseases and preventing them, an important thing to know about is vaccines. So how do vaccines actually work to prevent diseases? Well, a vaccine is a version of a pathogen that can't actually cause harm. So it's either dead pathogens or weakened ones or chemicals from a pathogen that'll trigger your body. So they trigger the body's immune system to be prepared to fight that pathogen. So that way, if the real pathogen comes into your body already has a defense prepared. And the chapter ends with the emerging diseases section. So emerging diseases are new threats, either previously unknown diseases that have appeared in a population, or diseases that were under control and then they change to become more dangerous. Those are both types of emerging diseases. So they talk about three different kinds. Superbugs are versions of pathogens that have evolved to be able to survive our medications. So these happen from overusing antibiotics and antibacterial products. Um, the idea is those products will kill almost all of them, but then there'll be a few left that survive, and they're strong enough to survive. They're the ones that reproduce, and then they make offspring bacteria that are also strong enough to survive. So superbugs are really scary, actually, because if they keep spreading, then we get these diseases that can't be killed by the antibiotics we have. An example of this is MRSA. It's a resistant strain of bacteria. So it's what's called a staph infection. There's staph infections that can be treated pretty easily, but MRSA is a version of it that's become a superbug. Our normal antibiotics don't work to stop it. Another example of an emerging disease would be viruses that infected one species, and then the virus evolved to be able to infect more species. So remember, viruses have to interact with the proteins on your cells, so normally they're pretty limited. There's only one species or a few species that they can infect. But as the virus evolves, it might be able to interact with different proteins. So examples of this would be HIV that once infected other primates but couldn't infect humans, but eventually evolved and became able to infect humans. And bird flu, which did the same thing. It affected birds, and now it can also infect humans. And the third type of emerging disease mentioned are called prions. And these are kind of weird. They're not really a bacteria or a virus, but they're misfolded proteins, and that can kind of build up and damage brain cells. And prions are spread when animals eat other animals that are already infected with them. So they do spread like other diseases. All right, so that's the end of the chapter. We've got two review questions. Same thing, we'll just read them, give a few seconds to think about it, and go over the answer. So what are antibiotics used for? For fighting viral infections only, bacterial infections only, both viral and bacterial infections, and fighting influenza? Only one of these answers is correct. I'll give you a few seconds to think about it. Okay, so hopefully you remember this one. Antibiotics are not used for fighting viral infections. It's bacterial infections only. So not viral infections, not both, and not the flu because the flu is a viral infection. Okay, influenza is always a virus. And our last one, we've got those emerging diseases. So we talked about superbugs, prions, and new viruses. So I'll give you a few seconds to think about which is which. All right, misfolded proteins that pile up in the body and cause problems. Those are prions, pathogens that evolve to infect more species than they used to. We just kind of refer to them as new viruses. And when antibiotics are overused and resistant pathogens evolve, we get superbugs. So that does bring us to the end of this chapter. So if you have any questions, don't hesitate to give us a call or send an email. We hope to hear from you soon. You have a great day.